Tricarboxylic acid cycle. Plan of the lecture: Bioenergetics, reactions of the tricarboxylic acid cycle, and the regulation of the tricarboxylic acid cycle. Bioenergetics. The main energetic currency in cells, our cells and cells of animals, bacteria, and plants, is ATP, adenosine 5' triphosphate. It contains adenine as a base, ribose, and free residues of phosphate group. Two of them contain high energy phosphate bonds. The free energy related when ATP is hydrolyzed is used to drive reactions that require energy. For example, transport of ions against of concentration difference between membrane. ATP can be hydrolyzed to form ADP and inorganic phosphate, or AMP and pyrophosphate. The unhydrolyzed bonds of ATP are often called high-energy bonds. ATP carries two high-energy phosphate bonds. ATP is constantly being consumed and regenerated, consumed by different biochemical processes which need energy, and regenerated by two main processes. The first one is glycolysis, and the second one is oxidation of different components like beta oxidation of fatty acids inside mitochondria. The energy transformation can be divided into three principal phases: oxidation of fuels, fat, fatty acids, carbohydrates like glucose, proteins, or amino acids, conversion of energy from fuel, oxidation of high energy phosphate bonds of adenosine triphosphate, or ATP, and use of ATP phosphate bond energy to drive energy required process like transport of ions, like catalyzing of different biochemical reactions. Reactions of the tricarboxylic acid cycle. The first reaction, which supply acetyl coenzyme A for tricarboxylic acid cycle, is a conversion of pyruvate into acetyl coenzyme A. This reaction needs many coenzymes like coenzyme A, NAD, thymine pyrophosphate, lipase, and FAD. After production of acetyl coenzyme A from acetate, from glucose via pyruvate, fatty acids or ketone bodies or amino acids, acetyl coenzyme A enters into tricarboxylic acid cycle, and after that, the transport energy electrons from acetyl coenzyme A onto electron carriers and like NADH or FADH. We transfer electrons using these carriers and transfer them onto respiratory chain. The cycle also serves as synthesis of fatty acids, amino acids, and glucose. The cycle starts with the 4-carbon compound oxaloacetate, adds 2 carbons from acetyl coenzyme A, loses 2 carbons as a CO2, and regenerates the 4-carbon compounds oxaloacetate. Electrons are transferred by the cycle to NAD and FAD, as the electrons are subsequently passed to oxygen by electron transport chain, and in this electron transport chain we produce a specific enzyme. ATP synthase produces ATP from inorganic phosphate and ADP. Describing the production of energy in tricarboxylic cycle or citric acid cycle, we have to keep in mind that our enzymes produce 3 molecules of NADH, which can produce 2.5 molecules of ATP, 1 molecule of FADH2, which works as a source of energy to produce 1.5 molecules of ATP, and 1 molecule of GTP in animal cells and ATP in plants and in several types of bacteria. Regulation and synthetic functions of the tricarboxylic acid cycle. The main idea of regulation of tricarboxylic acid cycle is related with concentration of ATP in cells. So the main function of tricarboxylic acid cycle is a supplying of free electrons, which are carried by NADH and FADH from different fuels to respiratory chain. The respiratory chain, this several sequence of proteins or enzymes, works as a carrier for electrons to oxygen, and after that a specific enzyme, ATP synthase, produce ATP from ADP and inorganic phosphate. ATP works not only as a source of energy for different biochemical processes, it works as a regulator for activity of tricarboxylic acid cycle, and it works as an inhibitor production of products of NADH and FADH to prevent accumulation of ATP inside the cell. Also, accumulation of ADP, adenosine diphosphate, works as an activator for tricarboxylic acid cycle, which activate production of electrons from fuels, from transport electrons from fuels to respiratory chain to regenerate ADP to form ATP. Synthetic functions of the tricarboxylic acid cycle is related to with production of different components, organic components, like fatty acids and steroids from citrate, which should be transported from a matrix of mitochondria to cytoplasm. Production of different amino acids from alpha-ketoglutarate, like glutamine, proline, arginine, glutamate, or from glutamate we can produce purines. Production of heme from succinyl coenzyme A. Production of aspartate, asparagine, and after that pyrimidines. Production of glucose from oxaloacetate, and at the same time an excess of phosphoryl pyruvate could be used as a source for production of serine, glycine, cysteine, phenylalanine, tyrosine, tryptophan.